Fumi, thank you so much for coming and flying across to speak to us.
Thank you, Dr. Barrett, and thank you, Tracy, and everyone at the Wolfram UK for inviting me to here. And it's always great to see patients from the UK, so nice to meet you. My name is Fumi Urano. I am from Washington University in the United States. And today I'm going to focus on our therapeutic development for Wolfram syndrome in the United States. So this is always I use. To achieve a cure for Wolfram syndrome, we have 3 steps. And our first step is to stop the progression of the disease using medicine. Second step is to protect and regrow remaining tissues. And third step is to replace damaged genes and tissues. And so for the second and third steps, it may take time. So our first step is to stop the progression using medicine. And so this is the mechanisms, and basically we believe that endoplasmic reticulum stress is the root cause of the disease. And we want to target the upstream of the mechanisms using a drug to stop the progression of the disease. So as all of you know, the downstream manifestations of the disease is the diabetes, insulin-dependent diabetes, visual impairment, and other symptoms. And upstream, very upstream, is the Wolfram gene mutations. And we want to target as upstream as possible. We want to target the upstream of the disease mechanisms. So what we are targeting right now is to target endoplasmic reticulum calcium depletion. So that's what we are trying to target. And so we have been trying to identify FDA-approved drugs that can target the upstream mechanisms of the Wolfram syndrome. So FDA is equivalent to EMA in Europe. And the reason is, as Dr. Barrett just described, we can relatively go faster to give these approved drugs to patients. So these drugs are approved for other conditions, but it's relatively easy to bring these drugs to patients suffering from different conditions. So we did compound drug screens, and we identified a drug called Dantrolene sodium that can potentially delay the progression of Wolfram syndrome. So what's Dantrolene sodium? This is approved by FDA. It is also approved by EMA for the treatment of different conditions. And this drug targets ryanodine receptor localized to the endoplasmic reticulum. So I believe Wolfram is an endoplasmic reticulum disease. Endoplasmic reticulum, a cellular compartment, executes many, many different functions. And this drug targets the endoplasmic reticulum. It is currently approved for spasticity, muscle stiffness, and it is also approved for the treatment of malignant hyperthermia. So some people are very sensitive to anesthesia, and then sometimes their muscle contracts too much. It creates huge amount of heat. And this drug is approved for the treatment of malignant hyperthermia as well as spasticity, which is muscle stiffness. And so this is how Dantrolene sodium acts. It acts on the endoplasmic reticulum. And we have different types of animal models of Wolfram syndrome. So the way we usually test candidate drugs in fast cell models, we have Wolfram gene depleted cells, and we discover Wolfram gene depleted cells, and we also created animals that lack Wolfram gene, and these are Wolfram syndrome mouse models. And the black is the so left hand side is control, and right hand left side is control, right hand side is the Wolfram animals, and black circle means some cell death genes, cell death molecule is activated, and by treating these animals, we could suppress the activation of cell death molecules. So red is treated animals treated with dantrolene sodium. So these are brain samples. So we also treated diabetes Wolfram mouse model with this drug. And so red is Wolfram animals, and green is the treated animals. So this is blood sugar levels. So we did a glucose tolerance test. Basically, we give sugar to Wolfram animal model, Wolfram syndrome animal model, and blood sugar levels went up quickly. And that's red, and so upper side. And so in dantrolene treated animals, blood sugar did not go up like very high as Wolfram animals. So that suggests that dantrolene sodium is working. We also looked at pancreatic beta cell mass. So in Wolfram animal model, there's reduction in beta cell mass, and we could partly restore beta cell mass by treating these animals with dantrolene sodium for 4 months. We treated animals for 4 months. We also looked at structures of these animals. So the beta cell structure is beta cells from Wolfram animals have a little different structure. This is control Wolfram animals, and beta cells from Wolfram animals. And by treating these animals with dantrolene sodium, we could improve the structure of the beta cells from Wolfram mice. We also we collected skin cells from our patients with Wolfram syndrome, and then we created pluripotent stem cells from these skin cells. And these cells can be differentiated into different types of cells, including brain cells, eye cells, and insulin-producing cells. So these are skin cell-derived stem cells. And we created brain-like cells using derived from skin cells of our patients. And these cells from patients are susceptible to cell death. This is cell death marker. So control and Wolfram cells, and the cells from Wolfram patients are susceptible to cell death. This is cell death marker. And by treating these cells with dantrolene sodium, we could partly, not perfect, but partly reduce cell death. So this is what we are doing right now. So we plan to conduct clinical studies, further preclinical studies in the Wolfram neurodegeneration mouse model using dantrolene sodium. But we could obtain orphan drug designation of dantrolene sodium for the treatment of Wolfram syndrome from US FDA early in 2016. So in February this year, we could get the approval. And so we plan to do a phase 1B safety studies in patients with Wolfram syndrome using dantrolene sodium. So this is the design of a clinical trial. So the clinical trial protocol was just approved by our Washington University Medical Center. And so this is going to be an open label and ascending dose study in both children and adult patients with Wolfram syndrome. So we will assess the safety and tolerability of dantrolene sodium administered orally at upper end of therapeutic dose range for 6 months. Then we will also assess the effects of dantrolene sodium on remaining beta cell function visual acuity, just like Dr. Barrett mentioned, and neurological brain functions. So this is the dose escalation protocol. So we will do a screening visit. Then every week we will slightly increase the dose of this drug. And for pediatric patients, we will create elixir. So they take the liquid form of Dantrolene. And duration is 6 to 9 months, 6 months treatment and follow-up. We plan to recruit 24 patients. We started recruiting patients last week, and the primary endpoint is safety and tolerability. So we will determine the dose for the further, longer period trial. We will monitor beta cell function, remaining beta cell function, visual acuity, and visual functioning questionnaire. And we will monitor the neurological functions using the Wolfram Unified Rating Scale. And we will, for this particular trial, we will not use MRI because this trial is short. And so this is anticipated timeline. So we just started screening and recruitment of patients, and we plan to invite first patient in November or December this year. And funding is coming from a Snow Foundation and also Very White Foundation. Stephanie Snow is here, so she actually played an important role to make this trial happen. And we try to get a little more money from National Institute of Health. It's pending right now. So this is a drug repurposing trial, just like Dr. Barrett mentioned. So this is a timeline. So we plan to conduct phase 1B, 2 clinical trials in the United States in between 2016 to 2018 or 2019. And Dr. Barrett's team will conduct trial from 2017, I'm sorry, then we are planning to conduct a joint phase 3 clinical trials, international multi-center clinical trials using the fund from Horizon 2020. So that's our plan, and things are moving forward. I understand we are not fast enough, but things are moving forward. And so to stop the progression, this is drug repurposing trial, but just like Dr. Barrett mentioned, we also need new drugs. So we are also developing new drugs. This is a collaboration with Washington University and the National Center for Advancing Translational Sciences in the United States. It's a government-based drug development institution. And I visited this institution together with Stephanie Snow Gable, who is over there. So clearly patient voice is very important. So we visited together and met with the director of this institution. And so the director was very impressed by the collaboration between patient organization and researchers. So it's very important to get your voice to move things forward. And so clearly there are 4,000 rare diseases and 7,000 rare diseases on this planet. So clearly this institution cannot work on all the rare diseases. So we are very fortunate to be picked. So we have been conducting drug screens with this institution, and we identified a few promising drugs. This is one of them. And so by Wolfram gene depletion induces cell death, so this is control. Number goes up by depleting Wolfram gene. And this drug can suppress the cell death induced by Wolfram gene depletion. This is even better than Dantrolene sodium. So new drug candidate, we have few new drug candidates, and so we are conducting medicinal chemistry. That means we are changing the structure of new drugs to make it even more potent, and we are conducting pharmacokinetics and toxicity studies. So we need to make sure that this new drug goes to eyes, brains, and pancreas. So this is so-called pharmacokinetics, and clearly toxicity is very important because we don't want to give any toxic drug to our patients. And we conducting preclinical studies in cell and animal models for the investigational new drug application. This means we need this to do a new clinical trial using these new drugs. And this is anticipated timeline, so hopefully we can complete toxicity and PK studies by the end of 2018, and hopefully by 2020 we can start phase 1 safety study.